Hello everyone, today I'll be talking about the physiology of the vision. This video is based upon the chapter 3rd from the Parson, which is the book of ophthalmology. So the outline of this chapter is: first there is a description about the rhodopsin cycle, then the magnocellular, parvocellular and the choanocellular pathways and so on. In this video we'll just be discussing the rhodopsin cycle. Rest of the topics will be discussed in the subsequent chapters. So now, as we all know, that the function of the eye is to form a clear image of a stimulus on the surface of the retina, right? Now the anterior surface of the cornea, it accounts for about 2/3 of the refractive power of the eye, and the lens, it further reflects the rays of the light which is entering the eye and is responsible for the ability to see objects at all the distances. The ocular lens changes shape. It is more spherical for the near object and flatter for the far ones. What happens is there is a relaxation of the ciliary muscle. So these are the relaxed ciliary muscles. So as a result of which the suspensory ligament attached to the lens, they become taut, and then the lens becomes thin, and therefore it is focused for distance vision. Now when the ciliary muscle fibers are contracted, the suspensory ligament are relaxed and the lens becomes thick, and it is focused for the close vision or the near vision. And sorry for this typing error. The photoreceptors are more than the retinal ganglion cell. So there are approximately 1 million retinal ganglion cells in the human eye, and there are approximately 125 million of photoreceptors. So at the fovea there is a 1:1 correspondence between the photoreceptors and the retinal ganglion cells. But what happens in the periphery? There are more photoreceptors. Sorry for this, my typing error. That there are more photoreceptors as compared to the retinal ganglion cells. So what happens is there is a progressive loss in resolution as we move towards the peripheral part of the retina. Now this is a photoreceptor. The signals are transmitted to the ganglion cells via a bipolar cell in between. So what the eye does, the eye converts the light energy in the wavelength of 397 to 723 nanometer into electrical signals in the optic nerve. A single rod is able to detect light with a peak sensitivity of about 505 nanometer, which is roughly the green light. And the light rays which is falling upon the optic disc give rise to no visual sensation. Hence this is called as the blind spot of Marriott. The light which is falling on the other part of the retina, it causes two essential reactions: the photochemical and the electrical. The photochemical changes, it concern the pigments in the rods and the cones, and the most explored pigment is the rhodopsin, which is basically a chromoprotein. It is made up of a chromophore, which is the 11 cis retinal, which is attached to the 3 bond to a large protein called as the opsin. So this is basically a chromoprotein, right? And now coming on to the rhodopsin cycle. The rhodopsin, what exactly rhodopsin is? It is a visual pigment in the outer segment of the rods, okay, and which is responsible for the scotopic vision or the ability to see in the dark. Rhodopsin has 11 cis retinal as its chromophore, which is embedded by 3 different chemical bonds into a single peptide transmembrane protein, which is called as the opsin. So what happens in the visual cycle or the retinal cycle? As the rhodopsin is exposed to the light ray, there is formation of photorhodopsin, hypsorhodopsin. They are formed almost immediately. Then meteorodopsin is formed in picoseconds. Lumirodopsin is formed in microseconds. Meteorodopsin 1 in milliseconds, followed by meteorodopsin 2, which activates transducin and produces all transretinal and opsin or the scotopsin. This all transretinal is converted to the vitamin A, and with the help of the isomerase enzyme, it is then again converted to the 11 cis retinal, and then these two are again combined and form the rhodopsin. So this visual cycle keeps on happening. A human rod cell can respond to a single photon absorption, demonstrating the efficacy of the photoisomerization. So if there's any queries, you can post them in the comment box, and in the next video I'll be talking about the magnocellular, parvocellular and the choanocellular pathways. Till then, happy studying.